What if I told you that um, the mushroom you just casually step over on a weekend hike might actually be an evolutionary parent to a completely different species on another continent?
Yeah, or, a victim of a decapitating cellular body snatcher.
Right, and an organism whose entire physical appearance is basically just an elaborate lie.
Yeah.
Today we are unpacking the uh, the biological horror, the genetic deception, and this massive global network of a fungus that most people have never even bothered to look at twice.
It really is the ultimate lesson in why we just cannot trust human observation. I mean, we wander through these forest ecosystems utterly blind to the brutal biological warfare, the symbiotic hostage situations,
Totally.
and the genetic fluidity happening just, millimeters below the soil. Like the macro world we see is quiet, but the microscopic reality is this chaotic, fiercely competitive metropolis.
And the undisputed protagonist of that metropolis, or, well, perhaps the central victim, depending on how you look at it, is this humble, straw-colored little mushroom known to science as Cystoderma amianthinum. Most foragers just call it the earthy powdercap. It has this tiny, granular, almost like mealy-looking cap. It is the absolute definition of unremarkable.
Yeah, it really is.
But beneath that unassuming exterior, the earthy powdercap is actively dismantling the fundamental rules of modern biology. Okay, let's unpack this.
Yeah.
Our mission for this deep dive is to tear through a massive stack of complex taxonomic research, genetic sequencing data, and global biodiversity compendiums. We are tracking this fungus from the dense, chilly needle duff of Newfoundland all the way to the high-altitude, arid scrublands of Pakistan.
It's quite the journey.
And to understand the scale of the deception we're dealing with, we have to start with the great illusion of the wrinkles.
Yeah, the morphological illusion is the perfect entry point, I think, because it really exposes the foundational flaw in how early scientists cataloged the natural world.
Right, because for decades, mycologists were out in the field studying the earthy powdercap, and they cataloged this very consistent, seemingly obvious physical difference.
Exactly. Some of these fungal fruiting bodies had perfectly smooth, rounded caps, but growing right alongside them, like literally in the exact same soil, were mushrooms with deeply ridged, heavily wrinkled caps.
Almost resembling like a topographical map or the folds of a human brain.
Right, it was a very distinct texture. And in classical taxonomy, form dictates function. If two populations consistently look different in the wild, the assumption is that they possess completely different genetic blueprints.
So taxonomists just drew a hard line in the sand.
They did. They split them into two distinct varieties. You had Cystoderma amianthinum, variety amianthinum for the smooth morph,
Okay.
and then variety rugosa which basically means wrinkled and netted for the textured morph.
And they published it in the field guides. Foragers meticulously keyed them out based on cap texture, and it was accepted as a hardened taxonomic reality.
Yeah, that is until molecular phylogenetics completely shattered the paradigm.
As it usually does.
Right. Let's look closely at a major molecular study utilizing fungal collections from Newfoundland and Labrador. The research team there didn't rely on visual morphology. They ground up the tissue from both the smooth and the wrinkled varieties, and they sequenced the ITS region.
Which is, it's the perfect tool for this specific job. We're talking about the internal transcribed spacer region.
Yes, exactly.
It's sandwiched between the highly conserved ribosomal RNA genes, meaning the spacer mutates fast enough to show us recent evolutionary divergence, but stable enough that it doesn't just degrade into total genetic noise.
Right. It's the gold standard barcode for distinguishing closely related fungal species. The resolution the ITS provides is unparalleled for this kind of species-level delimitation.
So what did they find?
Well, the researchers ran the ITS sequences for dozens of these earthy powder caps, and they were expecting the data to neatly bifurcate into two distinct clades.
Like one branch for the smooth genetics, one for the wrinkled?
Exactly. Instead, the resulting phylogenetic tree was just an absolute mess of intermingled sequences. A deeply wrinkled rugosa mushroom shared an identical ITS sequence with the perfectly smooth amianthinum growing like 3 inches away.
Wow. So the genetic distance was 0.
0. Variety rugosa was a phantom. It just did not exist at the molecular level.
So if the genome isn't coding for this radical structural difference, what is driving it? I mean, the fungus isn't just deciding to put on a disguise.
No, of course not. It has to be an environmental or a mechanical trigger acting on the cellular architecture of the cap. And it comes down to a mechanical failure of the tissue driven by cellular turgor pressure.
Okay, trigger pressure.
Right, and the sporocarp's aging process. When researchers cross-referenced the physical specimens with the environmental conditions at the time of collection, the actual mechanism revealed itself.
Which was what?
Well, imagine a fresh cluster of these mushrooms emerging from the soil after a heavy, saturating rain. Their cells are hypergorged with water.
Right, they're fully hydrated.
Exactly. That internal hydrostatic pressure keeps the cap tissue taut, plump, and perfectly smooth.
But mushrooms are incredibly ephemeral. I mean, they have no real mechanisms to trap or retain that moisture over time, do they?
None at all.
Right.
So as the days progress, or, if a dry wind moves through the understory, the moisture gradient reverses. The mushroom begins to lose water to the atmosphere.
Okay, I see where this is going.
Yeah, the internal turgor pressure drops, the hyphal cells physically contract, and the rigid surface area of the cap basically just collapses inward on itself.
Creating those deep, netted folds.
Yes. A smooth cluster of mushrooms on a humid Tuesday literally becomes the wrinkled variety by a dry Friday.
Wow. So early mycologists classifying these as different varieties is essentially like a space alien coming to Earth and classifying a grape and a raisin as two entirely different species of fruit, not realizing one is just a dehydrated version of the other.
That's a perfect analogy. The physics of the collapse are remarkably similar. The alien sees a plump, green, turgid orb and then a wrinkled, brown, dense object and assumes they're distinct evolutionary lineages.
But the underlying genome hasn't changed at all.
Right. Only the hydration state of the cells has.
Which highlights a terrifying reality for anyone trying to map the fungal kingdom.
Not right.
I mean, if the physical traits of the earthy powdercap are this malleable morphological taxonomy is essentially useless.
It really throws a wrench in the whole system.
But replacing the morphological lens with a genetic one doesn't actually solve the problem of the earthy powdercap, does it? In fact, it plunges us into an even deeper structural crisis.
Oh, absolutely. Because when you map the entire global genetic footprint of Cystoderma amianthinum, its actual place on the evolutionary family tree breaks one of the most sacred rules of biological classification.
Yeah, it forces a confrontation with paraphyly.
Yeah.
And paraphyletic evolution is the concept that basically keeps strict taxonomic purists awake at night.
It really does. Because in textbook evolutionary biology, the gold standard is the monophyletic group.
Right.
That's a clade where you have a single exclusive common ancestor, and every single descendant of that ancestor is neatly contained within that one taxonomic box.
Like the ultimate clean biological filing cabinet. The ancestral population splits, species A goes left, species B goes right, and they both become something new and distinct.
Exactly. But the genome of the earthy powdercap utterly rejects the filing cabinet.
Tell me about it.
When geneticists sequence the ITS region of this fungus across vast, geographically isolated populations, the genetic variation simmering inside the species is so wide, so messy, that it forms a paraphyletic continuum.
And to grasp the mechanism of this, we need to examine a rogue lineage isolated in the neotropics, right? Cystoderma and pinum,
Yes.
the Andean powdercap,
Yep.
found in the extreme high-altitude paramo ecosystems of the Ecuadorian Andes.
Yes. And geographically, ecologically, and morphologically, C. encynum is universally recognized as a distinct, valid species.
Because it's adapted to a totally different environment.
Right. It is adapted to high UV exposure, dramatic temperature fluctuations, and an entirely different suite of host plants.
But when its DNA was sequenced and placed into the phylogenetic tree,
Yeah, the data showed that C. encynum didn't just share an ancient common ancestor with the global earthy powdercap population. The genetics revealed that C. encynum derived directly out of the C. amianthinum lineage.
wait, really?
Yes, the Andean species acts as a basal offshoot, literally a genetic child of the widespread earthy powdercap.
Wait, if C. endinum evolved out of C. amianthinum, how can we still categorize C. amianthinum as a single cohesive species? Doesn't that break the fundamental architecture of how we name life?
It absolutely does.
I mean, if a subpopulation splinters off, migrates to the Andes, and genetically drifts into a new species, but the parent population in North America and Europe just keeps on interbreeding and remaining genetically static as the original species.
Yeah.
The parent species is essentially existing concurrently with its own evolutionary offspring.
You've articulated the exact boundary where human nomenclature fails to capture biological reality.
It's just wild.
It is. The earthy powdercap isn't a static genetic entity. It is an ongoing, localized evolutionary event. The sheer size and interconnectedness of the global C. amianthinum population means it acts as a massive genetic reservoir.
So a tiny splinter group can get isolated in the high Andes, perhaps, spores carried on high-altitude wind currents.
Yeah.
And that isolated group experiences a massive founder effect. They undergo rapid, localized adaptation and speciate into C. anthinum.
Exactly. But that speciation event has zero effect on the billions of earthy powdercap mycelia thriving in the boreal forests of Canada. The umbrella is leaking, but the umbrella remains intact.
It fundamentally changes how you view a species. It's not a finished product. It's a messy, bubbling cauldron of genetic potential.
That's a great way to put it.
And while its DNA refuses to be put into a neat phylogenetic box, its daily survival tactics down in the soil are just as defiant. To really understand the ecological role of the earthy powdercap, we have to look at its diet.
Right, because when scientists actually bring this fungus into a sterile lab and try to feed it, it effectively goes on a hunger strike.
The metabolic mystery of this fungus is arguably its most fascinating trait. Let's look at its preferred microhabitat.
Okay, so if you track the collection data, particularly in that extensive Newfoundland study where C. amianthinum constituted nearly 80% of all Cystoderma specimens.
Which is a huge majority.
Yeah, massive. They are almost exclusively found fruiting deep within moss beds and thick needle duff. They completely dominate this specific stratum of the forest floor.
Which would logically lead you to assume they are standard decomposers.
Right.
I mean, most forest floor fungi are saprotrophic. They secrete enzymes to break down the lignin and cellulose in dead pine needles, rotting wood, and fallen leaves.
And saprotrophs are notoriously easy to culture in a lab. You just drop their spores onto a nutrient-rich agar plate, and they devour the free carbon.
But the earthy powdercap rejects the agar plate.
It does. Researchers face an incredibly difficult, often impossible task trying to get the myospores, the sexually produced spores dropping from the dills, to germinate in pure laboratory cultures.
You can provide them with the perfect temperature, optimal pH, and a smorgasbord of complex carbohydrates, and the spores just remain completely dormant.
Right, and this prolonged, stubborn dormancy points away from simple saprotrophy and strongly suggests a biotrophic lifestyle.
A biotrophic dependency, meaning they require a living host organism to complete or at least initiate their life cycle.
Exactly. They aren't interested in the dead pine needles. They are waiting for a biochemical signal from something alive in the soil.
This brings us to the pioneering fieldwork of mycologists Heinemann and Thone in the 1970s, and later Harry Harmaja in the 80s.
Yes, they moved away from the lab and started meticulously excavating the root systems beneath the fruiting bodies out in the wild.
Which is tedious work.
Oh, incredibly. But they observed that the mycelial threads of the earthy powdercap were inextricably woven into the living rhizoids of specific mosses.
Particularly those in the genus Polytrichum, commonly known as haircap moss.
Right. So we are looking at an obligate symbiosis. The spore lands in the soil, and it enters a state of deep metabolic suspension.
It refuses to wake up until it chemically detects the specific root exudates the metabolic byproducts secreting from a living Polytrichum moss nearby.
That is the dominant hypothesis, yeah. The spore requires the moss's chemical signature as a germination trigger. This raises an important question.
What's that?
Well, regarding our fundamental understanding of forest ecology, how much of the fungal network we assume is merely breaking down dead matter is actually tied into a vast invisible web of living microscopic dependencies?
That's a staggering thought. The earthy powdercap might not just be growing near the moss, it might be parasitizing the moss.
Exactly. Siphoning off the sugars the moss produces through photosynthesis in an ancient biological hostage situation that we simply cannot replicate in a sterile Petri dish.
It is deeply humbling. I mean, we possess mass spectrometers and advanced genetic sequencers, yet this common brown mushroom refuses to perform unless we bring it its specific living moss companion.
It really puts our technological hubris in check.
It really does. But if the earthy powdercap is secretly operating as a moss parasite, the forest ecosystem has a brutal sense of irony.
Oh, absolutely.
Because the data shows this fungus is simultaneously the target of one of the most horrific cellular hijacking predators in the natural world.
The hunter becomes the hunted in the most visceral way possible. We have to examine the pathology of the Squamanita genus.
Yes, the Squamanita. These are incredibly obscure, highly specialized parasitic fungi species, like Squamanita paradoxa and Squamanita basii.
And they do not operate like typical parasites that simply attach to a root tip and passively drain nutrients. The Squamanita fungi are aggressive body snatchers.
I want to dig into the actual mechanics of this hijacking, because you occasionally hear about entomopathogenic fungi like cordyceps taking over the bodies of ants.
Right, the zombie ants.
Exactly. But this is a mushroom attacking another mushroom. It is a fungal-on-fungal crime. How does it physically manifest?
Well, for decades, the evidence was purely macroscopic. Mycologists would find these bizarre, deformed chimeras in the woods. They would discover a fruiting body where the base of the stem was grotesquely swollen.
Distorted into a dense tumor-like mass, right? Called a protocarpic tuber.
Yes, but the truly chilling aspect was the cap. Emerging from this deformed base would be a mushroom cap that morphologically belonged to an entirely different genus.
It is the biological equivalent of a sci-fi horror film. It's like an alien landing on you, decapitating you, and replacing your head with its own, while using your legs to walk around.
It is absolute fungal horror. The cellular reality is just as violent as your analogy. The Squamanita mycelium detects a developing earthy powdercap primordium.
The primordium being the tiny nascent pinhead of a mushroom forming under the soil.
Exactly. The parasite invades the host tissue. But it doesn't just consume it. It secretes specific biochemical compounds that effectively arrest the host's genetic expression for cap development.
Wow. So it literally decapitates the host at a cellular level.
Yes. It hijacks the vascular flow of water and chitin building materials moving up the host's stipe and redirects all of that biological turgor pressure into forming the Squamanita's own cap and reproductive gills.
That is ruthless.
The earthy powder cap is kept alive just long enough to act as a biological hydraulic pump, pushing the parasite's spores into the air current.
But wait, if the host's cap is never allowed to form, and the host's stem is deformed into a swollen tuber, how did mycologists ever definitively prove that the earthy powder cap was the victim?
It's a great question. Without the cap, morphological identification of the host is impossible.
Right.
Which is why the host identity remained a matter of intense debate until a landmark 2010 molecular study by mycologists Matheny and Griffith.
Okay, what did they do?
They recognized that morphology had hit a dead end, so they turned to microdissection and DNA sequencing. They harvested a fresh, chimeric specimen of Squamanita paradoxa.
The ultimate molecular autopsy.
Exactly. With incredible precision, they extracted DNA tissue from the alien parasitic cap at the top. Then, they took separate tissue samples from the deformed, hijacked tuber at the base.
And because the tissues are literally fused together, isolating pure host DNA without contamination from the invasive parasite must have been a massive technical hurdle.
Oh, it was. It required specialized primers to amplify specific target sequences. When they finally processed the data, the cap tissue predictably sequenced as Squamanita paradoxa.
As expected.
But the ITS barcode extracted from the hijacked, swollen stem returned a perfect match for our unassuming friend Cystoderma amianthinum.
Oh, wow.
Yeah. The genetic data definitively proved the earthy powder cap was the host.
It's just a staggering life cycle. The earthy powder cap lies dormant in the soil, waiting for the chemical signal of a living moss. It parasitizes the moss, begins to build its fruiting body, only to be ambushed, genetically silenced, and decapitated by a secondary parasite.
It is a brutal trophic cascade happening right beneath our boots.
But tracking these interactions, identifying who is eating whom in the needle duff, required mycologists to completely untangle the taxonomic mess of the Cystoderma family tree.
It did. And that effort culminated in what we can call the Great Taxonomic Split.
A necessary, if controversial, biochemical reckoning. To understand the split, we have to look at how the genus was originally built. In 1889, a Swiss mycologist named Victor Fayod established the Cystoderma genus based on a single macroscopic trait.
Which was the presence of a granular mealy veil covering the cap and stem.
Right. If a mushroom had that powdery coating, Fayod basically just threw it into the Cystoderma bucket.
It was a dumping ground built entirely on superficial visual similarity.
And convergent evolution proves time and time again that unrelated organisms can evolve similar physical traits if they occupy similar ecological niches.
Exactly. Relying on a mealy cap is a recipe for a polyphyletic disaster, a grouping of species with no recent common ancestor.
And that biological reality finally caught up with the genus in 2002.
It did. Finnish mycologist Harry Harmaja took on the monumental task of cleaning up the Cystoderma dumping ground.
Okay.
He realized visual morphology was failing, so he shifted the diagnostic metric to biochemistry. Specifically, he utilized Meltzer's reagent to test the complex starch composition of the spore walls.
Now, Harmaja using Meltzer's reagent is a classic mycological technique. The reagent is an iodine solution containing potassium iodide and chloral hydrate.
Right.
The mechanism relies on the iodine reacting with amylose-like macromolecules embedded in the spore's ornamentation. If those specific starches are present, the iodine forms a polyiodide chain, triggering a dramatic color shift to a dark grayish blue or blackish blue.
And we call those spores amyloid.
Amyloid, right? But dividing an entire genus based on whether spore wall starches form a blue-black complex with iodine? I mean, that seems like relying on a single, potentially fragile point of biochemical failure.
It is a high-stakes taxonomic gamble, for sure, but Harmaja leaned into it. When he applied Meltzer's reagent to true Cystoderma species, like our earthy powdercap, the reaction was immediate and strongly amyloid. The blue-black complex formed.
Okay, so they passed the test.
But a significant chunk of the mushrooms sitting in Fayod's original bucket showed zero reaction. They lacked those specific amylose macromolecules entirely. Their spores were in amyloid.
Wow.
Yeah. Based on this singular biochemical divergence, Harmaja violently split the family tree. The amyloid species remained in Cystoderma.
And the amyloid species were evicted.
Exactly. They were placed into a newly minted genus, Cystodermella.
An elegant binary system. Iodine turns blue, you go in box A. No reaction, you go in box B.
Right.
And the poster child for this newly created Cystodermella genus was the sister species to our earthy powdercap, a mushroom called Cystodermella cinnabarina, the cinnabar powdercap.
A visually stunning organism. It produces a vibrant, deep, reddish-orange fruiting body. Because its spores are resolutely enameloid, Harmaja formally moved it into the new genus.
But the moment modern molecular phylogeneticists began sequencing the DNA of Cystodermella cinnabarina to confirm its new placement, they accidentally uncovered a massive, paradigm-shifting genetic secret regarding its physical appearance.
Yeah, they really open a can of worms there.
Every time we shine a molecular light on this family of fungi, the concept of a fixed physical identity collapses. What did the DNA reveal about the cinnabar powdercap?
It revealed that C. cinnabarina operates as a highly fluid genetic chameleon.
A chameleon.
Yes, for decades, field mycologists have been cataloging other mushrooms that possess the exact same complex microscopic architecture as the cinnabar powdercap. Specifically, they shared distinct cellular structures called cystidia.
Cystidia. Those are sterile cells on the gills that, in this case, take the form of tiny, elaborate, crystal-encrusted spears, right?
Exactly, nature's microscopic armor. The structural similarities were identical, but the macroscopic colors were completely divergent.
Drastically divergent, yeah. Mycologists documented a mushroom with the exact same crystal spears, but its cap was a dull, earthy brown.
And following the morphological rulebook, they named it as a distinct species. Cystodermella myrtecystis.
And then they discovered an entirely albino, pure white variant with the same microscopic spears.
Right, and they categorized that as yet another distinct species, Cystodermella lactea.
Red, brown, and white. 3 distinct colors, 3 distinct Latin names.
Exactly.
Here's where it gets really interesting. The phenotypic plasticity of these organisms is so extreme that it routinely fools our best visual taxonomy.
It completely upends it.
When geneticists finally ran the ITS and large subunit ribosomal RNA sequencing for the red cinnabarina, the brown myrtecystis, and the albino lactea, the sequences laid perfectly over top of one another.
The genetic divergence was essentially negligible.
Wow.
They are not 3 distinct species. They are a single, highly adaptable master species, expressing wildly different color morphs based on environmental or localized epigenetic triggers.
It completely obliterates the utility of color in field identification.
It really does. You are essentially looking at the fungal equivalent of an Arctic fox, changing its coat from summer brown to winter white. But early mycologists assumed they were discovering entirely different branches of the mammalian family tree.
That's a great comparison.
What's fascinating here is that the genetic fluidity of Cystodermella cinnabarina proves that these fungi possess a vast library of unexpressed genetic potential, turning physical traits on and off as needed.
But the revelation of this master species brings me back to my skepticism about Harmaja's great split.
Okay, let's hear it.
If these fungi possess such immense genetic flexibility, if they can completely alter their pigmentation while remaining the same species, How much faith can we truly put in the iodine test?
Ah, I see what you're saying.
I mean, if nature loves a chaotic exception, there must be a species that breaks Harmager's rigid biochemical rule.
And your skepticism is completely validated by the existence of Cystoderma japonica.
Ah, there it is.
It is the ultimate rogue lineage, and it proves that biochemical litmus tests have massive evolutionary blind spots. When Harmaja evaluated C. japonicum in 2002, he dropped the Meltzer's reagent on its spores. The result was starkly an amyloid. No starch reaction, no blue color.
So, following his own strict protocol, he evicted it from Cystoderma and transferred it into Cystodermella.
Right. It obeyed the chemical rule.
But I'm guessing the molecular phylogenetics completely contradicted the iodine.
Entirely. Subsequent DNA sequencing of the large subunit and ITS regions placed C. japonicum squarely in the center of the core Cystoderma clade.
Wow.
Genetically, it shares a recent, undeniable common ancestor with the earthy powdercap. It is a true Cystoderma.
But somewhere along its evolutionary journey, a genetic mutation silenced the biochemical pathway responsible for synthesizing those amylose macromolecules in the spore wall.
Exactly. The trait was lost, but the lineage remained. It is a stark reminder that a single chemical test cannot map the true, messy complexity of evolution.
Spore wall chemistry is a valuable clue, but DNA is the ultimate arbiter of truth. And when we look at the DNA, the true scale of these fungal networks becomes apparent. We spent a lot of time zoomed in on microscopic crystal spears, localized iodine reactions, and cellular body snatchers.
We have.
But the genetic flexibility we see in species like the cinnabar powdercap directly translates to massive ecological adaptability.
Yeah.
These aren't fragile, hyperlocalized organisms. To truly grasp their success, we need to look at their global geographic footprint.
Their distribution is staggering, and there is really no better illustration of their adaptability than a recent monumental compendium of macrofungi compiled from the incredibly diverse ecoregions of Pakistan.
Right, and this research effort wasn't just about listing names. It was a comprehensive effort to map the biological infrastructure of an entire nation across violently contrasting climates.
Exactly.
When most people envision Pakistan, the immediate mental image is, the towering glaciated peaks of the Karakoram and the Himalayas.
Of course.
But the ecological gradient of the country is vast, ranging from alpine tundra all the way down to coastal mangroves. The compendium recorded a massive 12,293 species of macrofungi spread across 15 distinct ecoregions.
And right there in the data, thriving in these ecosystems are the lineages we've been discussing, Cystoderma amianthinum and its close cousin, Cystodermella granulosa.
Tracking exactly where they thrive within those 15 ecoregions provides incredible insight into fungal survival strategies.
It really does. The compendium data reveals a highly concentrated distribution. A staggering 36% of all recorded macrofungal taxa in Pakistan are clustered in the western Himalayan broadleaf forests.
Which makes perfect ecological sense.
Right.
A broadleaf forest provides a dense, continuous canopy that drastically reduces ground level evaporation. It generates a deep, complex litter layer of decaying deciduous leaves, creating the perfect humid microclimate for saprotrophic activity.
And supporting lush beds of moss for biotrophic relationships.
Exactly. It is a fungal paradise.
But the true testament to their evolutionary resilience lies in the runner-up ecosystem. The compendium notes that the second highest concentration of fungal diversity, hosting 25% of the recorded taxa, is found in the northwestern thorn scrub forests.
Now that is wild. A thorn scrub forest is the absolute antithesis of a damp Himalayan canopy.
Completely.
We are talking about highly xeric, arid environments defined by sparse, drought-resistant vegetation, intense solar radiation, and chronically dry, compacted soil.
Yeah.
How does a fungal genus that thrives nestled in the damp moss of Newfoundland survive in a harsh northwestern thorn scrub?
It requires extreme metabolic and morphological adaptation. In an arid thorn scrub, a fungus cannot rely on consistent surface moisture. The mycelial networks must plunge deep into the mineral soil to locate subterranean water tables.
Oh, wow.
Or they must develop highly specialized sclerotia-like resting structures, hardened masses of dense mycelium that can survive years of total desiccation.
So they just shut down.
Basically, they enter a state of extreme metabolic dormancy, waiting out the punishing dry seasons, only rapidly pushing up a fruiting body during brief, localized flash monsoon events.
So what does this all mean? We've covered a massive amount of incredibly dense biological mechanics today. We've talked about hydrostatic turgor pressure causing wrinkles, paraphyletic speciation in the Andes, obligate moss biotrophy.
The cellular decapitation by the Squamanita parasite.
Yes. And complex iodine search reactions
Yeah.
and survival strategies in arid scrublands. Why does this complex matrix of fungal data matter to the listener going about their daily life?
If we connect this to the bigger picture, it fundamentally alters how we perceive the resilience and architecture of the natural world. This compendium in Pakistan, much like the sequencing data from Newfoundland, proves that fungi are the foundational invisible infrastructure of our planet.
Right.
When we track the earthy powdercap across the globe, we aren't just tracking a mushroom, we are mapping the historical health and evolutionary adaptability of the Earth's ecosystems.
Because they're so deeply tied to everything else.
Exactly. These organisms possess the genetic fluidity to change their colors, silence their chemical outputs, and wait out decades of drought. Understanding their underlying mechanisms helps us predict how complex ecosystems will adapt, survive, or fail in the face of rapid global change.
The dirt under your boots isn't just dead rock and rotting leaves. It is a sprawling, intelligent, biological supercomputer, calculating survival strategies in real time.
Exactly that. It is ancient, patient, and endlessly adaptable.
What a wild, unsettling, and brilliant journey for one seemingly unremarkable brown mushroom.
It really is.
We started by stripping away the earthy powder cap's morphological disguise, revealing a fungus whose deeply wrinkled cap is nothing more than mechanical illusion brought on by a lack of hydrostatic pressure.
A physical trait driven entirely by a dry breeze, fooling generations of taxonomists.
Right. We looked deep into its genetic barcode, the ITS region, and found a paraphyletic anomaly, an evolutionary parent that remains genetically static in the boreal forests, while simultaneously birthing a completely new, distinct species, Cystoderma antimonum, in the high, punishing altitudes of the Ecuadorian Andes.
A species existing as an ongoing continuum, completely ignoring the clean, monophyletic boundaries we try to enforce.
And we uncovered the deep mystery of its metabolism, tracking a spore that violently refuses to germinate in a sterile lab, strongly suggesting an ancient, obligate, biotrophic reliance on the chemical exudates of living Polytrichum moss.
A hidden symbiotic tether that proves how much of the forest floor is interlocked in ways we still cannot synthesize.
Then we detailed the visceral horror of the Squamanita strangler fungus, a parasitic predator that doesn't just consume the earthy powder cap, but actively hijacks its vascular system, arrests its genetic cap development, and forces the deformed host body to disseminate the killer's own spores.
A literal masterclass in cellular subversion and localized biological warfare.
And finally, we watched the entire genus get split in half by the Melzer's iodine test, a biochemical reckoning that revealed massive genetic chimeras capable of changing their colors at will, and rogue lineages that abandon their starch chemistry entirely, all thriving across a vast global gradient from the humid Himalayas to the arid scrublands of Pakistan.
A true testament to the sheer, unyielding genetic flexibility of the fungal kingdom.
It completely upends how we look at nature. And as we wrap up this deep dive, I want to leave you with a final thought to mull over. We've spent the last hour meticulously dismantling the idea that you can trust what you see. We've proven that mushrooms painted in wildly different colors are genetically identical, while mushrooms that look perfectly identical belong to entirely different evolutionary lineages. We've seen a swollen stem reveal itself to be a hijacked hostage. So the next time you take a walk through a quiet, damp forest, the next time your boots crunch down on the decaying pine needles, ask yourself, how much of our understanding of biology, and perhaps reality itself, is heavily distorted by our own human visual bias? What other invisible chemical or genetic truths are actively playing out right beneath your feet, completely ignoring the inadequate labels we've placed upon them?